we talked about the different classes of hormones, and we said that the classes of hormones are going to be what? Based on amino acids, peptides, or lipid derivatives, right? All right. So peptide hormones are made out of what? Peptide bonds. What has peptide bonds? Proteins, right? Or linear structure of amino acids. Yes. So what are the peptide hormones? Well, peptides are hydrophilic, right? So peptide hormones are secreted by whom? Whom? Well, that is your adenohypophysis, your hypothalamus, thyroid gland, parathyroid gland, and pancreas. Before going into there, I'm going to give you a nice overview of this information. How do your body knows when to release what, and how to release it, and where to release it to? Question is, you have a bunch of sensors spread throughout your whole body, correct? And every single cell and every tissue. Whenever something is sensed, right, that's going to travel all the way, either through a negative feedback, or positive feedback, or a nerve impulse, right? This nerve impulse will trigger an information or trigger a process inside your brain, and your brain will say, okay, if my blood pressure is high, what do I want to do? I want to do the opposite. I want to take it to a lower level as long as it's within normal limits, yes? So then within your brain, which is, again, the first order will be your central nervous system, which is, again, your brain, which is, that's your processing center, right? Brain, spinal cord. And then within the brain, you have structures. So this is part of your brain, which is the second order will be your endocrine system. And this endocrine system possesses two units in here. Let's put it 2A and, I don't know, 2B. Means the brain will send impulses to a portion of the brain, which is known as the hypothalamus. And now this is just part of your endocrine system. And then the hypothalamus will send information, as you can see, either to the front or the back of your next gland, which is your what? Hypophysis. Hypophysis or pituitary gland. So B is either hypophysis or pituitary gland. Everybody learns pituitary. Nobody learns hypophysis. Well, guess what? Learn hypophysis, because that's the one in the test. Now, the hypophysis is going to be broken down into two parts. So this gland, first of all, is attached to the brain. So it's part of the brain. It's attached to the brain to this section right here, and that is called the pituitary stalk. And because of this physical binding, on the front of this gland is going to have is going to be called adenohypophysis. Why adeno? Because it has a glandular, and you can see there's a lot of blood vessels going all the way to that front of the gland, right? So anterior or posterior compartment of the hypophysis are known as adeno or neuro. Why do we call it neuro? We call it neuro because there is a direct neuronal connection. In other words, you remember from 285, you have a neuron which has a soma and a long axon all the way to dendrites. Well, those things you can literally see all the way here. You can see them, right? They're binding all the way here. So they're going from the hypothalamus all the way to your neurohypophysis, hence the name neuro. So adeno because it has a lot of glandular and blood flow, and neuro is because it has a bunch of neurons, direct neuron connection. So which one will act faster? The neuron, right? All right, awesome. So we got that part. Now you know which part is what and what are we talking about. So the next event that we're going to talk about is what? Well, adenohypophysis. The mnemonic for adenohypophysis, which I give on the lab already, is your flat PG. Can you read that there? Sometime. So flat PG, just imagine a flat P and a G. So to me it's P and a G flat. I don't know, something like that. It's kind of funny, right? I don't know. Whatever. So two letters sleeping. So flat PG. So a flat PG stands for what? F for what? Oh wait. Oh, I don't know my letters. Hold on. So F is for FSH. Then the next one is what? L for LH. A for ACTH. A for, well, ACTH, duh, you said it. And T for TSH. And then P for what? And G for what? Awesome. That means that if you know that mnemonic, the flappy G, you already know most of the hormones. You say like, I'm sorry, you're missing one. Like, yeah, I'm missing one, but that one is right here. Do you see that right there means? So this part is known as the parts intermedia. Do you see the part? You can sit there if you want. I'm not using my chair. So, parts intermedia, that one possesses what? MSH. What does MSH stand for? Awesome, that's what I want. I want you to struggle. Yes, I mean that way. So that is melanocyte stimulating hormone, which I already told you last class that you need to learn this. So that means that all these hormones that we just mentioned right now are what? Peptide hormones. Okay? Now, on the hypothalamus, which is in this case, you have a hypothalamus, you have an R. I keep putting the R. R stands for what? Regulatory or slash, you're right, releasing hormone. Because in other words, almost every single hormone that you have or you will have on either the, I have to touch it, on the adeno is growth releasing hormone. And it stimulates what? The release of the adeno to synthesize or create and then release the growth hormone itself. Does that make sense? So whenever you see an R, that comes from the hypothalamus. So hypothalamus is going to regulate the release of the hormones produced on the either anterior or posterior portion of the hypophysis. Everybody got that? All right. Now, the two hormones that are found on the neurohypophysis are these two, oxytocin and ADH. Okay? Ready? So I should leave it there, but you know me. Really quick, I'm going to go over each one of those. I can't stop myself. FSH is follicular stimulating hormone. It's going to stimulate the follicles on the female, and it's also going to stimulate the Sertoli cells on the male testes. In other words, that's for what? Gonads. Right? Either ovaries or testes. Okay? In the ovaries, what are they going to do? They're going to make you produce what? Your egg, lady. And for the guys, what are they going to make you do? Your spermatocyte, guys. So that is FSH. Now the LH works in the same way. Just that the LH is called luteinizing hormone, which stimulates on the female the corpus luteum, which is what is going to maintain your pre-embryo and embryo lives. Now on the male is going to stimulate latent cells inside the testes. At the end is to produce spermatocytes. Okay. Now the next one up is going to be what? ACTH, and that one is adenocorticotropic hormone, and that's going to go to the adrenal glands and the cortex. Okay. The next one is TSH, which is T for thyroid. So where's this hormone going to go? To your thyroid. The word says thyroid, right? So it's going to thyroid stimulating. So it's going to do what? Stimulate your thyroid to produce what? T3s and T4. Yeah, I get that you're talking about all these things, right? Prolactin and growth hormone, but where do they actually go on? You're saying it goes. It goes. Where does it go? Well, it goes to the organ, it goes to the tissue of the organs, but by what way? Does it use the express way or the regular lanes? No, it uses what? It uses your bloodstream, right? So it gets dumped into the bloodstream, and the bloodstream, as the heart pumps the blood, it just moves all the way through your body. Okay? So, because all these are endocrine hormones. Now, the other one peptides that we have is going to be your thyroid gland, parathyroid gland. So a thyroid gland produces calcitonin, parathyroid gland produces PTH, and then pancreas, insulin, and glucagon. We're going to do this once in the next 10-15 minutes. Okay. But these are all the peptide hormones. So if a question comes into play and they ask you what type of hormones are they, most likely what are you going to answer? Peptide hormones. Because there's a few what? A few lipid-based derivatives hormones. Okay. So what are those few ones? So a lipid ones. So lipid ones are your eicosanoids and your what? Steroids. Okay. So eicosanoids are what? Leukotrienes, prostaglandins, prostacyclins, and thromboxanes. Okay. I know they're weird words, but we're going to talk about at least for leukotrienes, right? And prostaglandins. Now, steroids. Well, these are the ones that your sex hormones are made out of. So what are your steroid hormones? Well, for sex, we're talking about what? Testosterone or? I don't see it. Whatever. Testosterone or estrogen. We also have estradiol. We also have aldosterone. It's a form of estrogen, but it's not truly estrogen. So aldosterone, which is for regulating sodium and chloride, so salts. Cortisol. Cortisol is what? Cortisol is a stress hormone. We also have prednisone. What is prednisone good for? Yeah, prednisone is used as a corticosteroid. Most of corticosteroids tends to be anti-inflammatory medications. They use anti-inflammatory medications. Okay? So, all that builds from where? From cholesterol. Okay? So those are your lipid derivatives. Before going into any details, let's understand what they actually do when they are secreted into the bloodstream. So, each one of these things, it has what? Each one of these hormones is is secreted into the bloodstream, but it has a specific target cell or a specific receptor, hence we call it target cell, right? Because it actually flows through the whole stream and is looking for that specific place that they can sit on or that they can activate something. And that place is when they have that receptor. So the target cells must have a specific what? If you don't have the specific receptor, that is not a target. Okay? The receptors of the cell can be either outside on the plasma membrane, or they can be what? Inside the cell. If they are inside the cell, where can they be? They can be either in the plasma, or they can be in the nucleus. Everybody got that? So we're going to talk about nuclear receptors or plasma receptors. That means as the hormone binds, if it's a lipid, where the lipid needs to bind to the cell membrane. Remember the cell membrane we said is a hydrophobic, right? Because it's a phospholipid bilayer. Any lipid is going to cross the cell membrane. So lipid hormones are faster to work on a cell. So lipids can go directly through the cell membrane and bind either to the plasma or to the nuclear receptors. You already got that. So those are lipid based. The rest of them, which is the majority of them, are peptide hormones. So peptide hormones, it needs to bind to the cell membrane. The cell membrane, when it binds, it opens a gate or a channel. The hormone comes in and then is able to do its effect on the inside of a cell. Okay, so there's a difference between peptides and lipids. Which is faster, lipids or peptides? What do they bind to? Lipids, what do they bind to? Testosterone, estrogen, what do they bind to? I just did it. It binds to either the plasma receptors, not the plasma membrane, the plasma receptors, right, inside the cytoplasm, right, or it binds to the nuclear receptors. So it directly binds to the stuff. Now, the target cell activation. How do you activate the cell or the tissue, or tissue or cell? So target cell activation depends on 3 factors. Oops. What are those 3 factors? Blood vessels. What is blood vessels? Well, it's the actual amount of blood, right? And the level of the hormone inside the blood vessel. Wait, I'm not following. Okay, which one will give you a sweeter drink? 1 tablespoon of sugar or 15? So 15, there's more what? Hormones, right? More sugars diluted into your drink. Does that make sense? I know it's a weird comparison, but if it does the job, it works. So these amount of hormones can be in the bloodstream as free. So how many of you are free? Free, you can do whatever you want. No? So if you have a boyfriend, girlfriend, or relative others, right, that you need to be responsible for them, that means that you're not free. Free is that they're not bound to anything. When you are bound, what does that mean? That you actually are attached. Physically, the hormone is attached to something else. How many of you are physically attached? No, you're sentimentally attached, but yeah. You get it. So when you're physically attached to somebody, that means that you're bound to what? Well, you're bound to things that are floating in the bloodstream. And those things are what? Steroids or thyroid hormones. Well, this bound can also be bound to proteins in your bloodstream, like what? Albumin, right? Albumin is actually the most frequent protein, or the most abundant protein in your bloodstream. So if you're bound to the protein, and this is pretty much how it goes, if I'm bound to this protein, I'm going to be sitting in the protein, and the protein is going to carry me around. So it's kind of like a carrier. The protein is going to drive me all the way to school. It's going to drop me at my class, and I do the stuff what I need to do at class. Once I'm done, right, I'm just going to get dismantled and leave. Or in this case, the protein will take me back. Does that allow me to interact with a lot of things? No, I'm just going to be specifically dropped where I need it to be dropped, correct? So when you're bound to a protein, your half-life, in this case, the function or the mechanism activity, stimulating activity, is going to last for a longer period of time. Because you're not bound into every single tissue as you go into the bloodstream. You're only bound to that specific tissue. Okay? Now, the relative numbers of receptors. What does that mean? If you have 2 receptors and 15 molecules versus 2 molecules and 2 receptors, which is the highest capacity? All right. Let me draw something here. So you have 1, 2, and 3 molecules. And I have 1 receptor versus 1, 2, and 2 receptors. Which one will have greater binding? A or B? Why? Because you got more receptors, 1 for each one. But what about A? Well, even though you may have more amount of hormones, what is wrong? Even though you have more amount of hormones, you are not going to have a binding for those. So the next one is the affinity of those receptors to the hormone. I don't know what happened here. Technology. Yeah, but I don't have the plug-in. All right, so the affinity for those receptors means that if you're actually bound to the receptors based on the structure that you have. So what does that affinity means? Okay, let's see. The point is that you have a receptor. Let's say the receptor looks like this, and you have another receptor that looks like that. But your molecule looks like this. So this is your molecule. And we're talking about, again, affinity. So which one will have more affinity? Receptor A or receptor B? So that's what we're talking about. The molecule has a shape and the receptors as well. So if you have a higher affinity for that receptor, that means you're going to take the place in there. Okay? So what are those three things again? The levels of hormones, the numbers of receptors, and the affinity of it. So what is upregulation or downregulation? When you upregulate means that you put more receptors, right, to respond to the hormones. Okay? What does that even mean? So if I send epinephrine down my body and I want to increase the heart rate, what do I need to do to the receptors? Increase them or decrease them? Increase them, right? So now epinephrine can bind to more receptors and I have more tachycardia or whatever it is that I want to have, right? Or my body decides to have. Now, downregulation means what? Pretty much the same thing, but I am already tachycardic. I still have a lot of epinephrine in my body, right? Adrenaline in my body. Would I want to maintain my heart rate increase and my blood pressure increase? No. What do? I downregulate my receptors. What do? I hide the receptors or just lose the receptors. So that means even though I have a lot of adrenaline, I cannot bind to the receptors, so my tachycardia and my blood pressure does what? Go down. Do you get it? Okay. So out of the two, which one is positive feedback?
First one.
Right. Out of the two, which one is negative feedback?
Down.
Right. So downregulation means negative feedback is if your blood pressure is high, what do you want to be? Lower. So whatever is causing your blood pressure to be higher, what are you going to do? Lose whatever receptors are binding to those. So you decrease the numbers of receptors. Okay? Wow, a lot of words in just three things. So decrease the numbers of receptors. That's what it means. Yeah, number sign. I'm sorry. You're right. So this is a negative example of negative feed. Okay, here, perfect. Noun. Decrease the numbers of receptors. Okay. Everybody got it? Any questions? All right, awesome.
Could it be like an upregulation also a negative feedback?
Yeah, an upregulation can be a negative feedback as well. So yeah, if you have, let's say for example, you have low amounts of the hormones, but you still want to maintain the effect, your body will put just more receptors. Okay. So interaction of hormones and target cells. Oh, that's a good example. So the example is testosterone as you age. So as male age, the testosterone, and females age, the estrogen levels decreases, right? But your body's going to try to put as much receptors as possible on the surface of the body, on the tissues. Like, I need the estrogen, I want to be beautiful, and I still want to be young. And then for the guy, be like, I still want to be muscular, I still want to have my physical maintenance, right? And then your body, it's just going to decrease the amount of testosterone, estrogen, and sadly, that's what happens. So that's an example of putting up more receptors. All right, so interaction of hormones and target cells. All right, so there are three types of hormones interaction. What are the three types and how the hormones are going to interact? It's going to have a permissive effect. Permissive means you allow other things to happen. So one hormone cannot exert its effect without another hormone being present, without another. What is permissive? You need both. All right? So a synergistic means what? Synergistic, that means that they work together, they produce the same effect. I'm just highlighting the keywords. Now, the antagonist means, well, the antagonist of you, it's the opposite. So the action is opposed. Okay? So three type of hormones interactions. And now, let's dump the hormones into the bloodstream. As you see, the free hormones in the bloodstreams, in this case, they're hydrophilic. If they're hydrophilic, what type of hormones are they? Peptide. So amino acid base, peptide base, protein base, right? It's all the same thing, right? They're just hydrophilic. So as they are hydrophilic, once they go into the tissue and they find a receptor, and here's the receptor in the plasma membrane, not in the cytoplasm, so this is on the surface. If it actually has a higher affinity for the receptor, it's going to do what? is going to bind to it. Once it binds to it, every single peptide, amino acid base, or protein based hormone is going to stimulate a cascade of event. All right, the cascade of event is known as should use another color, second messenger activation. So any amino acid, peptide, or protein base is going to stimulate a second messenger. All I'm doing is I'm binding to the membrane of a cell or any part of the membrane, and I'm saying, hey, relay the message for me. So the hormone is the first message. The second messenger is then the receptor that is sending the information into the cell. So far so good? So that's a second messenger. Now, what are the structures or what are the other proteins that are going to be used in able to relay the message? Imagine this is just like a race, one of those races, right? That you give the cone to somebody else, right?
The baton.
So regulatory protein is going to be stimulated by the second messenger, and the regulatory G protein is going to regulate if the message is going to go in or not. So the way I remember this one is Mr. G. Mr. G is the bouncer. You guys know what a bouncer is, right? When you go to the club, I said this, to the club. When you go to a club, you got the bouncer, right? And Mr. G, you hand him $100, and Mr. G says, "My man, VIP." So that means that what? Mr. G needs to get paid. Who's getting paid? Protein G. With what? Money? No. How do we pay? How do we pay inside the cell? ATP, energy. So the protein, the regulatory G protein, needs to be stimulated by what? By energy, by ATP. So once ATP stimulates the regulatory G protein, then Mr. G allows the second message to go through inside the cell. Why do we need this happen? We need for this to happen because all this message is going to target at the end what? Receptors in the nucleus, and it's going to tell the nucleus what? Well, that nucleus nothing. Who cares about inside the nucleus? We care about what? DNA. We're going to stimulate the DNA to produce what? Hormones. Let me give you an example. We talked about TSH, correct? We said that TSH was what? Thyroid stimulating hormone. Was it a peptide? Yes or no?
Yes.
Yes. So if thyroid stimulating hormone is a peptide, well, actually, I just picked the one that is at the exception. This is a peptide, but it's an exception. T3 and T4, they do not go to the receptors. Let me pick something else. All right, whatever, GH. All right. So once you secrete GH goes into the bloodstream. GH is going to bind now to the receptor. It's going to stimulate the second messenger. And at the level of DNA, what is GH going to do? I don't know. The GH is going to do what? Well, if this is DNA and this is my liver, it's just going to tell my liver the DNA of my hepatocytes or the liver cells to produce what? Insulin-like growth hormone or insulin-like growth factor, which is another hormone. So the DNA is going to be coded to produce another protein, right? Or amino acid base that is going to work as a what? As growth hormone. Does that make sense? Because I know we're talking about hormones, but hormones are no more than what? Group of peptides, group of amino acids, or group of proteins. So all you're doing is coding the DNA to produce a specific protein to act as another hormone. Everybody got it? So that's what we're stimulating the DNA to do so. Okay? Again, don't worry about not understanding the insulin-like growth factors like that. I'm going to talk about it later. Okay? So all you're doing is stimulating DNA to produce another protein. Now, on the other hand, you have this. Transport proteins. So if you're a transport protein, that means you're bound to a protein. If you're bound to a protein, you're hydrophobic. But if you're bound, what's going to be your half-life? Smaller or greater? Your half-life is going to be greater because you're bound to actual protein. What protein would that be? I already said albumin, right? Most likely that's what you're going to bond to. Everybody get it? No get it? All right. So you have a car. Car does what? Transports you, right? from point A to point B. That's a transport protein. Walking, well, that's you free. You don't have a bike either. So you gotta walk from point A to point B. Which one will keep you safest? Walking or car? Well, nowadays it depends. No. If it's raining, which one will keep you safest? If you are in an accident, somebody's trying to grab you or, I don't know, whatever, the car. If you have sun hitting on you, which one is going to protect you? The car. So that's why protein lasts longer. That's a really weird but effective way of comparing. So once you do that, then of course, if it's hydrophobic, okay? So hydrophobic means, do you have to bind to the receptors on the surface? No. You bind to receptors where? On the plasma membrane or receptors to the nucleus. Everybody got that? So your action is a lot faster because all you're doing again is what? Stimulating the DNA to produce another hormone or to secrete a specific things. Okay? So we're going to go into a little more detail about that second messengers. Okay? So, as mentioned before, we have intracellular receptors. Okay? And these intracellular receptors are going to just activate your genes once they bind to the nucleus, and they're going to stimulate transcription and synthesis of what? What we said, other proteins, which becomes later on other hormones. And that happens with who? Steroid hormones or thyroid hormone. Remember TSH, which is the T3 and T4, that's the exception. So these are hydrophobic hormones. Just those two, steroid and thyroid hormones, or the main ones, right? Apart from your testosterone, estrogen, right? Cortisol and all that. Now, the other hand that we have is the hydrophilic one, which are going to bind to what? Membrane receptors. If they bind to the membrane receptors, they're going to stimulate what? A second messenger, as mentioned before. That second messenger is going to be what? Your Mr. G, right? Protein G. And that stimulates your protein kinase. So once you activate it, that G protein, which is a regulatory protein, will activate a protein kinase. Will make it active because you just given what? ATP. And that protein kinase is going to do what in there? All you're doing is what? Opening an ion channel. In other words, if I am binding to the surface and I'm saying, hey, would you please open the door? You stimulate a second messenger, Mr. G. Mr. G is going to say, hey, dude, open the door for her. He's not going to do anything. He's just relaying the message. So the door will be open, and now my hormone, the hydrophilic one, is able to do what? Go through the cell membrane into now the cytoplasm, right, or the nucleus, and bind to the receptors. So far so good? So that's all you're doing, opening channels or door, okay? So you're opening an ion or stimulating an enzyme to do the function of entering, okay? So I'm gonna skip over this one. So, mechanism of hormone action. Normally, hormones are produced more of the following cellular changes. So, hormones, what do they actually do? They alter plasma membrane what? Permeability. What is plasma membrane? Guys, the cell membrane, right? Didn't we say that all they want to do is get through the membrane? Either by stimulating second messengers and opening ion channels, or by just directly going through them? Yeah, so permeability, right? They affect the permeability of the plasma membrane. They stimulate what? Once they get to the DNA or to the nucleus, what are they going to stimulate? Protein synthesis, right? That's what we said. And if they're going to be what? Either activation or deactivation of enzymes. Let's say, for example, you have too much T3 and T4, right? What would you want to do? Increase it or decrease it? Decrease it. So that will be inhibiting of an enzyme that is making already more T3 and T4. You got it? So that's a deactivation enzyme, or a stopping enzyme, if you want to call it that way. We also going to stimulate secretory activity, and of course, mitosis. What is mitosis? Cell division, right? Let's say, for example, I want to produce more testosterone. What do I need? Guys, if you want to produce more testosterone, what do you need? More stimulation, and definitely you need tissue, right? What do you produce testosterone? Testes? You produce testosterone in your testes? So if you don't have tissues, if your testicles are really small, that means that the capacity to produce testosterone should do what? Should do what? Decrease. If your testes tends to be bigger, then you have a greater capacity, as long as you have the proper what? Stimulus, right? So people, don't laugh, people that actually take growth hormone and testosterone, right, to enhance the physical looks, that chisel look that you all seems to agree with on the new Hollywood movies, right, and stars, they take growth hormone and testosterone, males, to do what? So they can become bigger or skinnier and they change their body whatever they want, right? So all we're doing is, if you do that in a constant way, chronically, you are affecting the testes to produce what you already produce by nature, which is what? Testosterone and spermatocytes. Does that make sense? That's why even though when you do injections of growth hormone, you do it in what? In cycles. You don't do it constantly because that can shrink your testes. And if you do it for a long period of time, even though in cycles, it will shrink your testes. You have smaller testes, less capacity for testosterone, so you depend on the exogenous of testosterone, and you are going to have probably less amount of spermatocyte, viable spermatocytes. All right? All right, the next thing, we're going to talk about that when we go into reproductive. So, this is weird. I'm going to keep it simple. Let's go for it. Okay? So, effects of cyclic AMP. We said that cyclic AMP is part of the what? Second messenger, right? The G protein. So what happens with the G protein? I'm going to make this bigger. So you have hormone, first message, binds to the receptor, G protein, second message, first, second message, and then that's going to stimulate what then? That's going to stimulate my adenyl cyclase, because I'm giving it what? ATP, I just mentioned. And then that's going to open ion channels or activate enzymes. Same thing that we did, just now in a graph. On the other hand, you can do what? Instead of activating, you can do what? Inhibiting. We said what? You can stimulate an enzyme, or you can stimulate a stop enzyme or inhibit the enzyme, right? So we're doing the same thing. Hormone, protein, the G protein, cyclic AMP, and all you're doing is what? Reduce enzyme activity. So in one end, you could stimulate the enzyme. On the other end, you do what? Inhibit or decrease the enzymatic function or the production of the hormone that you're already doing. So far, so good? Now, there are two different types of second messengers. One is through G protein and adenyl cyclase, which is this one that we did. And the other one is calcium. Why calcium? Let's talk about it. So the first thing, again, we have the hormone, right? Nothing new, same thing. And then we have the G protein. Yes? That is the same thing for both of them. What's changing? What's changing is what? You're opening now calcium channels. Okay? And now, you see calcium on this one, on the first one that we did, it was not there. All we're doing is now we're getting a lot of calcium to rush inside the cell. So the inside of the cell becomes more what? Positive. So it kind of like depolarizing the cell. So calcium is going to stimulate another enzyme in here, which is calmodulin, and that's going to allow an enzyme to be active. So, summary, you have G protein and cyclic AMP. And this one you have what? The same G protein and is calcium / calmodulin. If you want me to put it on a table, I'll put G protein here. Hold on. So I'll put G protein. And there are two ways of the G protein, right? The second messenger. Either say cyclic AMP or calcium calmodulin. Does that makes it better to understand? And these are all second messengers, okay? Now, which hormones do what? Look, on the bottom, I put it for you. If you have epinephrine or norepinephrine, you use all of them. Means which one? Both. So epi is in both circumstances. But which one is for what? Well, almost all of them work on what? In here. I'm not going to go into detail. I'm just going to give you the shortcut, right? Which is only few working here. Which is the one that works on calcium calmodulin? You have what? Oxytocin. What else do you see there? Regulatory. What is regulatory coming from? Your hypophysis, right? And then what else do you have? Eicosanoids. Everything else, all rest is what? Right here. So that's a summary of that. If you remember it, you get the question right. If I put PRL or TSH, what do you answer? What second message do you use? Cyclic AMP. If I put regulatory hormone, what do you use? Calcium calmodulin. If I put epinephrine, both. That's it. You get it? If I put oxytocin, calcium calmodulin. Which second messenger way? Calcium calmodulin. Straight up? All right. Any questions before I move? I know this is not weird, that's why I put that stuff in there. Time? Yeah, so the rest of it is here, effects of calcium levels. You can read on that. The rest is that one. You can read that one too, right? And then we're left here, all right?